Hi, this is your instructor Kwamishen. For the last part of our lecture on microbial genetics, we will look at mutation. So for mutations, mutation is when we're referring to a permanent change in the arrangement of the nucleotides in the genes of an organism. So when the sequence changes, it's definitely going to influence the type of messenger RNA that is synthesized and the subsequent protein that would be synthesized as a result of the messenger RNA. Now, the changes that happen due to mutation has some implication on the survival of the organism, whether it loses certain functions or not. And for microorganisms, it may contribute to their pathogenicity. We know that natural selection acts on variability, the differences that exist because of the differences in the genes that are expressed among different species. But since mutations occur randomly, natural selection just acts on these traits that are expressed. So for instance, if you have these beetles, you have brown and green, and you have a bed that favors the green, then eventually you're going to have a lot more of the brown surviving and being able to pass on their genetic constitution to the next generation. So we can have chromosomal mutations where the entire chromosome experiences some permanent changes to its structure. And that could be a deletion, so that you have A, B, C, D, F. This is the centromere. But in this case, because of mutation, the B has been lost. So the genes that are here where the B is on the chromosome will no more exist in this particular instance. You could also have a duplication. So in this instance, you have this section that has been duplicated. So B section has been duplicated in the chromosome. You could have an inversion where, if we cut through this, you would have AED, you would have FBC. So AED, FBC. So it has been inverted. Instead of having ABC, CB is over here and it's turned the other way. Now that. Then you could also have a translocation where there is a break, and so this break here, so this would then join this, but there's a break here, and so this segment will then join over here, and this other segment will continue to join over here. So you have ABC, JKL, GH, and then you have ID, EF. So these are examples of chromosomal mutations: a deletion, duplication, inversion, or translocation. Then for these mutations, how are they expressed then? If you have a duplication of chromosome 21, as happens in Down syndrome. Down syndrome is also called trisomy 21, where there's a duplication of the chromosome. Then you end up having 3 of the chromosome 21 instead of 2. And that duplication is a result of about 500 to 800 extra genes that have been added. Phenotypically, it leads to various expressions. So it results in developmental delays and physical disabilities as well. Some of these people who suffer from Down syndrome also have a higher chance of suffering from congenital heart defect. Now, individuals may also suffer from chromosomal mutations that are not duplications. So you have deletions that do occur. So deletion in chromosome 5, chromosome 4, chromosome 11 all lead to different types of syndromes and a spectrum of effects that we see phenotypically in these individuals. Then you could also have a gene mutation. So you've talked about chromosomal mutation. You could have a gene mutation. The gene mutation may be a point mutation or a frame shift mutation. For a point mutation, we're talking about a change in a single base pair. So we also call it base substitution. And so, for instance, if you have a DNA sequence like this is the original sequence, and then there is a mutation, and so instead of thymine here, thymine is substituted by cytosine. This is an example of a point mutation. It could lead to a missense or a nonsense, and we'll talk about that more in the next few slides. Now, for frame shift, what frame shift is this. Remember, the nucleotide sequences are read in threes. So you have 1, you have 2, you have 3, and so on and so forth. You could have a deletion or an insertion that would change which 3 nucleotides are read together. So there's a deletion here. You have 3, and then you have the next 3. If you compare this, the second, to this, you see a change there. Then you have CTG, and then you have CGC because of the deletion. So this is an example of what we call a frame shift. There has been a shift in the frame of the 3 nucleotides that are read together. Now, point mutations may happen naturally during replication when the DNA polymerase or the RNA polymerase, the DNA polymerase, because we're talking about DNA duplication. And so DNA replication, when the DNA polymerase is not able to do the proofreading. Remember, proofreading is done after DNA replication to correct any mistakes that may happen. If it's not done properly and there are still mistakes there, those would typically be point mutations. Okay. Note that they don't always lead to a change in a phenotype. They don't always lead to a change in a phenotype. And the reason is because the genetic code is redundant. The genetic code is redundant. So you have more than one codons coding for a particular amino acid. So even if there is a change in the particular nucleotide base and you have a substitution, it could still code for the same protein because there is redundancy built into the genetic code. If you have a nucleotide change, that doesn't necessarily lead to a new amino acid sequence. That's an example of a silent mutation. So there's a mutation, but there isn't really any effect on the organism because there's still the same protein is synthesized. And then there could be a change that results in a change in amino acid. And that's an example of a missense mutation. But even for those missense mutations, they don't always lead to phenotypical changes. So I'm trying to pictorially show you how these different point mutations are present. So you have a silent mutation in this instance where GGC codes for glycine, and there is a substitution of C with A, but it still leads to the production or the synthesis of glycine because of the redundancy that is built into the genetic code. So in this case, that's an example of a silent mutation. There's a mutation, but there is no effect, phenotypically or functionally, on the organism. Then you could have a missense, where you have an insertion. So you have ATA. It should rather be AAA. So this is lysine. You end up with a new amino acid, isoleucine. And that is an example of a missense. Then there is a nonsense mutation. A nonsense point mutation will lead to a stop codon. So you have TAT, but there has been a mutation. So instead of T, there is A. There is A here, and that leads to a stop codon. So the protein is truncated earlier than it should. That's an example of a nonsense point mutation. Then there is a read-through mutation. For read-through mutation, where there should be a stop codon, you have a mutation. So TAA, you have instead of TAA, you have TTA. So you have T replacing this A, and that means that the protein synthesis will continue instead of it stopping. So you have a longer than necessary protein that is synthesized. In real life, practically, how does these changes affect organisms? So we take humans, for instance. We have people who suffer from sickle cell anemia, and so the shapes of their red blood cells, you have a number of them having the sickle shape instead of this circular or globin shape. Now there's a protein, there's a gene that codes for the globin protein, which is in hemoglobin. So there is a gene, a globin gene, and this is the normal globin gene. This is a mutated one. What you would see is that there has been a flip, so that T comes to this strand, okay, and A comes to this strand in the mutated, and this is what leads to a sickle cell. It affects the protein that is eventually synthesized, and then the shape of the cell is affected as well. So for the RNA, you would have the regular one. For the mutated one, you would have this instead, and this will lead to a different protein, valine. This will lead to glycine. So two different proteins that are produced, and people with sickle cell anemia have lower than normal oxygen being carried around the cells of the body. Then there could be an insertion. An insertion could lead to a frame shift. So you have CCC. T timing is inserted in front here, and so you have 3 codons that are red, and then now you have T there. So T and these 2 are red, and then this will then join these. So it leads to what we call a frame shift. It affects the proteins that are synthesized downstream. So the proteins that are downstream are all affected. Then you have an insertion as a result of mutation that doesn't affect the proteins downstream. So this doesn't lead to a frame shift. This is a non-frame shift mutation. And for a non-frame shift mutation like this, you would have an entire codon being inserted. The 3 nucleotides are inserted. You still have these codons together. You still have this codon together. And so apart from this protein that changes, the others remain the same. So not all deletions or insertions will lead to a frame shift mutation. Another example, you have a wild type DNA, the one that occurs naturally, and there's a mutant one. What you see happening is that this also is an example of a frame shift mutation, because there is a change here. So you have G deleted, so it's deleted. And once it's deleted, now TTT would then be read together. So you have these three read together, and these three will be read together. So there's a change in the frame shift, and it affects the proteins downstream. Similar one here, you could have a frame shift that will lead to a stop codon, and when that happens, you have a shorter than normal protein. So an insertion or deletion of base pairs that may produce a stop codon downstream, and usually a shortened protein. It's an example of a frame shift mutation. So I want you guys to try your hands on this exercise here. So if you have an original DNA sequence, and can you produce a messenger RNA? Of course, and its amino acids. So use a genetic code to do this. Then you have a mutated version of the original DNA. So go ahead and synthesize a messenger RNA and the proteins as well. But more importantly, be able to interpret what's going on. If there is a mutation, of course there's a mutation. Is it a point mutation or is it a frame shift mutation? And if it's a point mutation, what kind of point mutation was it in terms of the effect on the protein? Was there no change? Was there a change in one amino acid? Or was there a premature stop signal? And the same applies to if it was a frame shift as well. So try your hands on this. If you like, you can join me on in office hours and see whether you're on the right path on that. But you have substances that may be chemical agents, or maybe radioactive agents, waves, that may contribute to these mutations in the genes. So if they're chemical in nature, they are called chemical mutagens, and they could lead to specific base pair changes. There are instances where you have altered DNA because you have a nucleoside here. That's this. It's as if this is adenine, but this is not adenine. It's a different nucleotide, but it looks very much like adenine. The difference is in the fact that it pairs with a different nucleotide. So adenine in the DNA would pair with thymine, but in this instance, in this mutagen, adenine, the mutated one, will pair with cytosine, and this would then be passed on to the offsprings as well. X-rays and UV radiation could also cause mutations, and that's why there are some restrictions as to the levels and extent that we get exposed to these radiations, because they could lead to damage in the DNA. So there's a destruction of segments of the DNA. They could also lead to damage, and for UV lights specifically, they end up causing thymine dimers. So whenever the thymines are close to each other, they will form a double bond between themselves, which shouldn't be the case. So you end up forming a dimer when they should be monomers, because they are nucleotides. So you have these agents that are examples of mutagens. We can actually measure the rate of mutagenesis, the rates at which mutagens are produced by different agents. Note that mistakes rarely happen during DNA replication. In prokaryotes, you have between 1 and 1 million to 1 in 1 billion base pairs before you can see any mistake. In eukaryotes, it's a bit more common, so you have 1 in 100 to 1 in 1,000, but that's still a lot of base pairs before you see mutations. And it all comes down to how big the gene of the organism is. So prokaryotes, based on their cell, their sizes, have a lot more genetic components than eukaryotes. But what the mutagens do is that they will significantly increase the rate at which these mistakes happen in the DNA. You can check the presence of mutagens, let's say antibiotic resistance, by having a media that has a particular antibiotic in it, growing a bacteria, trying to culture bacteria that you suspect to have a resistance to the antibiotic in it. And if it does have a resistance, it's going to grow on it. So that's an example of a positive selection. If you have another bacteria that you believe has lost a particular trait, let's say its ability to synthesize an important protein, histidine, potent amino acid histidine, what you can do is to have a medium that has no histidine in it and see whether it's going to grow on it. So that's an example of indirect selection. We do that using what we call replica plating. In replica plating, so you have a medium, for example, we use the histidine example. We have a medium that has histidine in it. We grow or culture the bacteria, and then we use a particular surface. You have this velvet sterile surface that you then use to press against the surface to pick the microorganisms. Then you transfer them to separate plates. One would have histidine, one would not have histidine. So you have two of these. Each of them you use to plate one of the plates. You need to note where you're beginning the plating, where your hand is, because the arrangement of the colonies as you have them here, as you transfer them, should be the same for the two plates so that you can compare directly. So after you've transferred them, you can then incubate and then check for the growth of the colonies. In this instance, you have a histidine containing medium where you see a bacteria grow here, but in the medium that has no histidine, there's no bacteria here. So what that means is that this bacteria doesn't have the ability to synthesize its own histidine, and so it only grows in the presence of histidine, where the histidine is already available, and then it's able to pick it up and use it. You also have carcinogens. Carcinogens and mutagens are not necessarily the same. You have certain agents that are mutagenic, and then they are also cancerous. They can also cause cancer. So there are certain mutations that are not cancerous in nature. We call them benign, and there are others that are cancerous in nature. We can also check whether a particular mutagen is carcinogenic or not, and we use what we call the AMIS test. The AMIS test. The idea is to use a particular bacteria that is a mutant and will need a particular compound or element in order to grow the normal strains. So let's take histidine again. So let's say we pick bacteria that needs histidine to grow, and you have a medium that has histidine in it, so they're able to grow here. And in this instance, organisms that are able to synthesize their own histidine and those that are not able to synthesize their own histidine will all grow. However, if we should expose these organisms to those that do not have the ability to produce their own histidine, we subscribe them or expose them to the suspected carcinogenic element or compound, and we see these number of growth happening on the medium, what it then tells us is that there has been a change in the genetic composition of the microorganism, so that they have become histidine positive, and that they are able to, in this case, spontaneously, because of the change that happened, they're able to grow at a faster rate than normal. So this is normal growth. This is faster than normal on a medium that they shouldn't be able to grow that. So that tells you right away that something is amiss in these strains of bacteria, and that they may be the elements or the substance that we expose them to may be carcinogenic. There's gene transfer that happens generally in microorganisms, and most of them would occur vertically from parent to child during cell division. However, you have a very small percentage of them happening horizontally when microorganisms pass the genes to other microorganisms that are not their offspring necessarily. It's less common, but it does happen. And the ways that this can happen are three forms, either by transformation, by conjugation, or transduction. Transformation may be natural or may be induced using chemicals. Natural transformation was first demonstrated by Griffiths, and this is what we call the Griffith experiment, where you had a mouse that was injected with colonies of bacteria that had capsules, and so were very virulent. And so once they injected them with these capsules, capsulated bacteria, they died. We're able to isolate the bacteria from them and grow them on a culture media. Then you have other strains that are not encapsulated bacteria and so are not virulent. And so if these were injected into living mouse, healthy ones, they continue to remain healthy, we're still able to isolate these unencapsulated or non-encapsulated bacteria from them. And then you have heat-killed encapsulated bacteria that were injected into mouse, and they continue to remain healthy. We're not able to isolate them because they were killed. They were heat killed. However, now in these two instances, in this instance where you have non-encapsulated bacteria and heat killed encapsulated bacteria, the organism lived. But if you combine these two together and inject them into a healthy mouse, the mouse died. So what really happened here? We're able to isolate encapsulated bacteria that were virulent. Remember, the ones that we introduced were heat killed, but we're able to isolate those that were actually encapsulated and living. And this is a result of transformation. The transformation has happened, and that is when DNA is transferred from the dead encapsulated strain to the non-encapsulated strains that are living, and then they end up becoming virulent themselves. This procedure would happen in a number of bacteria naturally, so Bacillus, Haemophilus, Neisseria, Streptococcus, Staphylococcus. These groups of bacteria are able to carry out gene transfers using transformation naturally. You can also do this artificially, chemically induce them when the bacteria is at a stage where you can actually pass this genetic material into them after disrupting their cell membrane or the walls, their cell walls. So you have transformation that could happen where you have a particular gene of interest, you disrupt the cell wall of the bacteria, it's able then to pick up the gene of interest, and some of it is incorporated into its plasmid, and the rest disintegrate. Now you have genetically transformed this particular cell. Then there is conjugation, where you have in conjugation there is a need for a particular plasmid. It has to be mediated by plasmid. In addition to that, in conjugation there should be direct cell-to-cell contact, and the two bacteria, one should be a donor and one should be a recipient. So it requires different mating types, a donor and a recipient. You need a sex pili. In E. coli, This has extensively been studied, and for the first time, it was the first time that it was observed that there is a fertility plasmid known as the F factor, fertility factor, which is needed to transfer this plasmid from one cell, which is the donor, to the recipient cell, which doesn't have the plasmid. Okay, so at the end of it, both of them then become positive for the F plasmid. You can also have transduction, where you have a bacteria that has been infected by a virus. Bacteria that is infected by viruses that infect bacteria, are called bacteriophages. Now, when a virus infects a bacteria gene may then be incorporated into its genome, and this can be transferred to other bacteria that the virus infects. The process is like this. So you have a cell, and you have a virus that has infected a bacteria, so we call it a bacteriophage. Bacteriophage will then inject its genetic component into the donor cell, and then the DNA that it's injected into the donor cell will then be incorporated into the protein synthesis machinery of the cell, DNA replication machinery of the cell. And then you have several copies that will be made, packaged into bacteriophages, and then the cell will then lyse, break apart, and then they will go ahead and infect subsequent ones. So this is a virus DNA, virus genetic material, and there's a bacteria genetic material, which will then be incorporated into a new bacteria here. So that's another way that genetic material or information is transmitted. I have here for you a plasmid map. So for plasmids, you would have a section that makes it selectable, and so they are introduced into the cell, and it makes it easier to select them whenever you need to. And then you could also have in plasmids transposons. Transposons are also called jumping genes. So these are segments of the genome that are able to mutate. So it creates itself and puts itself in different parts of the plasmid or the genome, and by so doing changes the genetic identity of the particular organism. And it does happen in organisms every now and then as well. There are different types of plasmids. Plasmids will replicate independently. They are not necessarily essential for life, but they may confer on these organisms pathogenicity, and that may help them to actually survive. There are types. In the types of plasmids, we have fertility, resistance, and virulence plasmids. And so they all have different roles that they play in the survival of the bacteria. So this brings us to the end of our lecture on microbial genetics.